Welcome back, ladies and gentlemen. We are going to continue our work through biology, starting with our molecular genetics unit. So this is video 5-1, and today we are going to be really focusing and honing in on the structure of DNA. Now, we've talked a lot about genetic code and how genetic code is the way that proteins are produced inside of your body. These proteins can range from really just about anything. It really determines who you are as an individual, who you are as a species, what particular traits you have, what characteristics you have, what you look like. So DNA is very important in determining who you are and really what you are. So we're going to take a look at the structure of this amazing molecule found deep inside each and every one of our cells. So hopefully by the end of this video, you should be able to understand the structure of DNA and also be able to explain how the strand of DNA, how each one is complementary to each other. You'll see what that means here in just a moment. Let's take a look at some key vocabulary terms that you need to make sure that you know for this video. We're going to start with nucleotide, dive into nucleic acids, deoxyribose, ribose, adenine, thymine, cytosine, guanine, double helix, and hydrogen bonds. So, long time ago, well, not really that long ago, a little more than 60 years ago, Maurice Wilkins showed Watson and Crick an X-ray from Rosalind Franklin. So back during this period of time, we knew that there was this material that's called DNA. We understand that it's the genetic code, but we don't really know a whole lot about its structure or how it really functions inside of the body. And a very famous duo by the name of Watson and Crick used this image from Rosalind Franklin to help discover and understand the double helix structure of DNA, as you can see in the picture here on the right. So what we do know is that DNA exists in two strands that go in a double helix or spiral staircase type shape. Now, while Watson and Crick get much of the credit, Franklin is frequently overlooked for her contributions, which is very unfortunate. Now, Watson and Crick would go on to win the Nobel Prize for this. However, Rosalind Franklin did not win anything, unfortunately, for her contributions to the discovery of the structure of DNA. So what do we understand and know about the structure of DNA? Well, DNA is composed of two strands of repeating nucleotides. Each nucleotide contains a sugar, a phosphate, and a nitrogen base. If you remember from our biochemistry unit, we talked about how nucleotides are the monomers of nucleic acids. One of the key differences between DNA and RNA is the sugar that's present. DNA has a deoxyribose sugar, and RNA has a ribose sugar. So as you can see here on the picture on the right, we have a bunch of repeating nucleotides. There's a few differences between them. You'll notice that some have C, some have A, some have G, and some have T. And we'll talk about the importance of those here in just a moment. But again, it's really key to notice how they repeat. Notice that the backbone, or the two sides of the ladder, like the rough things that you would hang on to, are made up of sugar and phosphate. And you have sugar, phosphate, sugar, phosphate. The little circle represents a phosphate. The little pentagon represents a sugar. And those repeat themselves. The nitrogen bases, however, the rungs of the ladder, are very different from one another. And we'll explore that here in just a moment. But as you look at those strands are put together, and then it takes its double helix shape when the two strands bond with one another. Now, you'll notice that there were a variety of nitrogen bases that were in the middle. There are four key nitrogen bases: adenine, thymine, cytosine, and guanine. And there are certain rules on how these pair up in a molecule of DNA. A will always pair with T, and C will always pair with G. This is known as Chargaff's rule. So you could determine the percentages of A, T, C, and G based on the percentage of one. So if I know I have 20% of A, well, A always bonds with T, so I'm going to have 20% of T. And you can either do other calculations to calculate C and G. These molecules are held together by hydrogen bonds. So if you take a look at the structures, the hydrogen is attached to the oxygen there in adenine and thymine, while another one, a nitrogen, is attached to a hydrogen through a hydrogen bond. Also notice that the number of hydrogen bonds differ based on the pair that's present. A and T only hydrogen bond twice, while C and G hydrogen bond 3 times. So think about which chemical bond might be stronger, A and T or C and G. Just something to think about. So as we mentioned before, DNA is found in two strands in a double helix or twisted ladder type structure. and each strand is held to one another utilizing hydrogen bonds. Now, the reason they use the hydrogen bonds is that they can actually be easily broken apart. We're actually going to need to access the nitrogen bases A, T, C, and G when we perform DNA replication or protein synthesis. The repeating nucleotides also make up the steps of the latter, so you can see how the structure of DNA is appropriately formed. Now, when we look at the orientation of DNA, the term that's used very frequently is anti-parallel. So they're parallel because they're like parallel lines. They never intersect with one another. But they're considered anti-parallel because of the orientation of the phosphates and the sugars. We use terms 5' and 3' to discuss this. So if we look on the left-hand side of the molecule of DNA, the 5' end is towards the top and the 3' end is towards the bottom. If you look at the opposite strand of DNA, the 5' end is towards the bottom and the 3' end is towards the top. All that means is that we just have strands that essentially run in opposite directions. Think of it like a road. On the right-hand side of the road, if you're in the United States, that's the way you travel up the road. On the opposite side, the left-hand side, traffic travels down. So you can kind of get an idea of what the term anti-parallel means and how that looks and applies to the concept of DNA here. So really the whole focus of this video is just for you to understand the basic structure of DNA. And keep in mind that DNA is a nucleic acid. It's one of the 4 macromolecules we've previously discussed. It is double-stranded, it is a double helix shape, and is composed of many repeating nucleotides. Each nucleotide contains a sugar, a phosphate, and a nitrogen base. And 4 different nitrogen bases exist in DNA. A pairs with T, and C pairs with G. Hopefully you understand a little bit more about this video and understand how DNA works. We'll continue our work on this with replication, protein synthesis, and mutations to round out our unit. Have a great day, guys. We'll talk to you later. Bye-bye.